Hi everyone, my name is Coral Addison, and today we're going to be talking about how scientists determine the genetic code. The first experiment that began to crack the code was done by Nirenberg and Matthaei. Their goal was to determine what amino acids are specified by codons made from one type of base. First, they added in a poly-U, or in other words, an RNA strand composed of only base U. Then a cell-free extract was added containing all the components needed for protein synthesis. Lastly, the 20 amino acids were added, and one of them was tagged. From here, the sample was incubated at 37 degrees, and sometimes later, proteins were made. Then the sample is filtered. And as you can see, the proteins are collecting on the filter, and now it is time to test for radioactivity, which in this case is positive. The proteins that were made in this experiment were radioactive. This is because the amino acids that were radioactive in the beginning were specified by the poly-U. However, it's not always this easy. For each of these codons, the procedure must be completed 20 times, and in each, a different amino acid must be tagged. Then this process is repeated for poly C, poly G, and poly A, concluding the results for this experiment. For Nirenberg, knowing what these polycodons coded for was not enough. What about all the other codons out there? What amino acids do they code for? And this is where a second famous experiment comes in, completed by Nirenberg and later. The goal of their experiment was to determine what other matches between codons and amino acids exist through the help of tRNAs. First step is to add a trinucleotide. Next, we will add in a ribosome. And finally, 20 tRNAs with amino acids were added, and one of the amino acids was tagged to be radioactive. And all of these components were mixed, and what happened was the trinucleotide bound to the ribosome, and the matching tRNA was also bound. The next step is to filter. The filter was large enough to allow unbound tRNAs to pass through. However, the filter would not pass any bound to a ribosome. Now it's time to check for radiation, which in this case, yes, radioactivity was detected. That means the amino acid was radioactive, was the one that was bound to the ribosome, meaning that the trinucleotide we used was the one that codes for this amino acid. Now you might be thinking to yourself, what if no radioactivity was detected? In this event, we would know that the amino acid was bound to the tRNA was not the tagged one. This means we need to try a different amino acid and try again. Now if we repeat this experiment for different codons, what we came to discover was our current genetic code. This information allowed us to complete the organized table that helps geneticists and genetics students all over the world